All right, so we're going to end today with our keynote address, and I'm going to let Stephen introduce someone who needs no introduction. But I'm going to hand the mic over to Stephen to introduce Scott Halstead, who will be presenting remotely, but then presenting a pre-recorded keynote address, but then will be available for a Q&A discussion afterwards.
What he just said. So our next speaker, which I guess they'll be pulling it up. So our next speaker is Scott Halstead. I don't think anyone has worked longer on dengue than Scott. My calculations is it's over 62 years. He's going to talk a little bit about it, but he has been singularly focused on this effort and trying to reduce the global dengue burden since he first started investigating with the Army why children in Southeast Asia were dying of this new disease. And he's been a mentor and an educator and a friend to many of us, and we wish he could be here, but technology will help us, even if he can't be here in person, to at least hear his words and his wisdom. So if I can get this to work. Okay, here we are. I'll push it one more time.
It's a pleasure to be here, and I thank Adam for doing the mechanics to get me on the screen so I have a chance to talk to you today. I'm sorry to miss the meeting in Syracuse. Look, we're talking about a big problem. I know the world is full of them, but dengue really is on the list. Next slide. I'm going to approach this fairly short review of what's wrong with dengue and what we need to do about it by starting with some dates to give us some perspective on how long we've been at this. Next slide. And I'm going to start right now today. And the reason why I use the alarmist terms is that this is the number of dengue cases reported to date by PAHO for all the countries in the Americas. This is a number that's nearly 2 times higher than any other that's occurred in the last 30 or 40 years. So we're breaking records. Next slide. And the question is why? What the heck is going on? And these are the couple of things that I've triggered, that I've identified. And in the course of the talk, we'll come back to them. Zika, as you know, invaded the New World a few years ago. There's the ever-looming phenomenon of antibody-dependent enhancement that enters into every calculus of dengue disease and dengue burden. And then, even though they're what I would call vaccine-like things, we really don't have an effective vaccine. And why is that? I regard the component of the Takeda vaccine, the dengue 2 live attenuated vaccine, as being the only authentic dengue vaccine on the market today. Next slide. So here we see Zika, this amazing thing that we have actually no really good idea how or why it arrived, but there it is, and if it came once, it can come again. But it did something really amazing that I think we don't yet understand. I think we have glimmers, but I don't see that there's a mechanism to really make effective serosurveillance on a continental scale for the Americas. And in some sense, we need that because these viruses, both the 4 dengue viruses and Zika, are affecting the entire southern continent of the Americas. Next slide. So let's look more specifically at Brazil. And here is Zika entering in 2015 and peaking in 2016. These are simply reported cases. You can guess that there's an enormous overlapping, an enormous number of total infections. There's always a mix of inapparent and apparent. So these are the apparent infections that actually got into the reporting system in Brazil. So for the first 2 years of the Zika epidemic, there was no impact on dengue cases. But by 2017, you can see that dengue cases were reduced by 75%. And again in 2018 by 75%. And then by 2019, which is 4 years after Zika arrived, dengue was back to its sort of a normal level. And here at the bottom is the number of cases in Brazil. In 2024, you can see 9,000 cases and 4,800 deaths. So again, the question to ask is, what the heck is going on? Does the introduction of Zika have anything to do with this very large number of dengue cases that occurred this year? Next slide. So now I begin the dates, and so this will give those of you who are new to dengue some idea of the perspective. We're dealing with a regional epidemic of dengue hemorrhagic fever that we can attribute probably to World War II. And so this phenomenon of a huge mix of the 4 dengue viruses and it's causing a pandemic dengue hemorrhagic fever is a relatively new phenomenon in human medicine. By sheer chance, the war was conducted in Southeast Asia, so the army was there and had established bases, including in Bangkok. And so in September 1962, I was sent from Walter Reed in Washington to Bangkok to establish the virus department in what was then called the CITO Medical Research Laboratory, now called AFRAMS. And I just put one little statistic at the bottom. When I arrived in 1962, the peak age of admission to the hospital for dengue hemorrhagic fever was age 3. And a very large number of cases occurred in infants. So we'll talk more about infants later. Next slide. Now, we put together a large hospital-based study and an epidemiological study. And within a few years, we had the data that showed that these severe cases of dengue in children accompanied their second of the 4 dengue virus infections. There were all 4 viruses circulating in Southeast Asia at the time. But in addition to that, and this is very important, infants, who were 8% of all hospitalizations in the early years, their disease occurred during primary infection. So a secondary infection is not critical to the outcome. Something else is going on. And these findings were reported at the Asian Pacific Science Congress in Tokyo. Unfortunately, the Asia Pacific Science Congresses, which once upon a time were an important venue for communicating science, have pretty much disappeared. Next slide. Okay, so September 1972 is actually a fairly important date in the history of dengue. This is when the Armed Forces Epidemiology Board and Walter Reed started dengue vaccine attenuation and ultimately a vaccine development. And we were in the mix, and so I applied. And in order to prepare for a contract, I took the 4 different dengue viruses and grew them in the existing tissue cultures that were used to support live attenuated dengue vaccines. And we found that all 4 dengue viruses grew in primary dog kidney cells. So we submitted a contract and were awarded a contract to develop a dengue 4 vaccine. And we had an interesting experience with that vaccine because we had subjected it to 35 passages in primary dog kidney cells, by which time we thought it was attenuated. But the Army wanted us to finalize the vaccine by passing it in fetal rhesus lung. So they wanted to have a standard substrate for their vaccine. But we discovered that just those few passes in a primate cell line actually restored the pathogenicity. So let's go to the next slide. The next important date is October 1979. Now this is 13 years after I went to Thailand and discovered that dengue was severe dengue was a secondary dengue infection, but we didn't really understand the mechanism. And in 1979, we reported into the literature the result of an experiment in monkeys, where we gave monkeys IgG dengue antibody, actually from a mother, a human cord blood antibody, and then infected the animals with dengue 2. And the animals that were circulating dengue antibodies had significantly higher viremias than control animals that had a human antibody but not dengue. So this is the formal proof that of the phenomenon antibody-dependent enhancement. Now that just establishes that it exists, but it doesn't explain exactly what's going on. Next slide. So what do we know about antibody-dependent enhancement? And I wish I could say we know everything, but we really don't. But these are some of the things we do know. That it's a phenomenon that you can demonstrate at the cellular level in vitro and in vivo. And if you look at an infected cell that's been infected with an immune complex, an IgG immune complex, the infection occurs in the presence as opposed to the absence of antibodies. And they occur in cells that have Fc receptors, monocytes and macrophages. And the type of antibody is an IgG antibody that attaches to the microbial surface but fails to neutralize. Next slide. And these are two of the schemes that show the nature of, or the history and nature of antibody-dependent enhancement. On the left-hand side is the evidence of enhanced viremia in a very large group of monkeys that I had infected in 16 different sequences, 1, 2, 1, 3, 1, 4, 2, 2, 2, 2, 3, 2, 4, and so on. And you can see there was a very significant increase in viremia in the animals that were infected in the presence of antibody. And then on the right-hand side, you can see the curve that you can derive in the laboratory if you sample a collection of monocytes that are infected over a period of time. Next slide. Now, it's important to understand that antibody-dependent enhancement can be almost a passive thing. In other words, immune complexes floating around in the bloodstream can more easily attach to the surface of cells than just the virus. So an immune complex gives a virus a slight increase in attachment frequency on cells. But intrinsic aid is something very, very different. Once the immune complex has entered the cell and initiated infection, the production of viruses per cell is greatly increased. And this is due to a suppression of antiviral innate immunity in the cell. So these this phenomenon has been described and was discovered actually outside of dengue, but it still is not perfectly well understood. But anyway, so intrinsic aid is an immune complex, an FC receptor suppression of antiviral uh, immunity. Next slide. Now, it's really important for everybody who's working on dengue to understand that this is not just a phenomenon of secondary dengue infection. Next slide. This is, as you can see, a perfectly lovely, I think, a Vietnamese child with 6 months old with dengue hemorrhagic fever. She has a large liver, which is diagrammed on her abdomen, and she looks right fine there, but this is the type of infant that comes in. They tend to be very healthy and come from good home environments. But the problem is they got mosquitoes and they have dengue infections. And if their mothers have transferred, as most mothers in Southeast Asia do, transferred huge amounts of dengue antibodies, not just to one virus, but to many viruses, these babies are at high risk of developing a severe disease, which ends in shock and death. Next slide. Now, this just illustrates the types of cells that you can study in vitro, monocytes, macrophages, and dendritic cells. But as you can see, it has to be mature dendritic cells and not immature dendritic cells. Next slide. And this is just the kind of results you should get if you're studying ADE. When you look at reports in the literature that claim to measure ADE, when you look at reports that are studying the ADE phenomenon, many times they just take a single outcome measure, and they don't often even explain the dynamics of the infectious process. So this is what's going on. You can see that this is a collection of mature dendritic cells that have been infected in the presence of antibody, and this is the array of antibodies that occur in the antibody containing cells and in the controls. Next slide. This is an important thing to remember. Next slide. Now, there are a lot of phenomena that occur with human antibody-dependent enhanced dengue infections that are mysterious, and this is one of them, and that is that as the interval between the first infection and the second infection increases, the severity of the ensuing disease increases. I had a chance to work in Cuba for many years, and in Cuba in 1981, in Havana, they had a dengue hemorrhagic fever outbreak caused by dengue 2, and Havana had a citywide outbreak of dengue 1 that occurred in 1977, which is 4 years earlier. And then the Cubans got busy and mostly eradicated Aedes aegypti, and there was no more dengue in the country until 1997. And that it occurred only in the city of Santiago, the far east of Cuba. But again, the 1977 dengue 1 outbreak had occurred in Santiago, but this new introduction of dengue 2 occurred in 1997, an interval of 20 years. And you can see that the dengue hemorrhagic fever case ratio was 7.6-fold higher, and the death rate was 25-fold higher. Wow, what's going on? Next slide. I think right here in Syracuse, you have a an author here that showed us that if you look carefully at children in Thailand who've had dengue, had Japanese encephalitis, and then experienced a dengue infection, that infection is made a little bit more severe by the fact that they're circulating a related flavivirus antibody. And the same thing has been shown with Zika. So we get back to the Zika phenomenon that we talked about at the beginning. And next slide. Another interesting phenomenon with the dengue story is that ethnicity alters the risk to severe dengue infection. Asian and Western Europeans are at high risk, but sub-Saharan Africans are at reduced risk. This is something we learned from Cuba, where you have a mixed population of Caucasians and sub-Saharan blacks. Next slide. We believe that the genetic community, which is this is what this is reflecting, is actually due to Africans being bombarded for millennia by yellow fever infections. So I think there's a very high expectation that this black population in Africa would have developed intrinsic genetic yellow fever resistance, and this has been studied in the Cuban population, but it's a phenomenon that still needs further understanding. Next slide. And interestingly enough, this increase in severity, which we really initially observed at these long intervals, is actually something that occurs on very short intervals. So in the Santiago outbreak, which itself was an interval of 20 years from the dengue 1 infections, there's actually a month-to-month increase in dengue hemorrhagic fever severity. But the interesting thing is that it's linked to changes in NS1. Next slide. So this is the amino acid change that occurred in the NS1 molecule and that were NS1 gene and gene expression in Cuba. Next slide. So what's going on with dengue? Why is it so lethal? Next slide. Well, as everybody knows, you had somebody working on NS1 in your group. NS1 and endotoxin have interesting analogies, and these analogies were pointed out by the Paul Young and the Ida Harris groups in USCF. And NS1 is a protein that circulates at high levels, having been released from infected cells, and it does a lot of things. Next slide. But what the Young and Harris groups have shown is that NS1 activates human endothelial cell monolayers, resulting in leakage, and it is indeed an endothelial toxin, and that makes dengue an NS1 toxic shock syndrome, and by itself one of the most interesting phenomena in microbiology. Next slide. So let's just get back briefly to Zika, which entered Brazil in 2015, and as we discussed earlier, it created a major epidemic in 2016, and then has declined to, it hasn't left the country, but it's declined to sort of low-level endemicity. And as we talked about before, the 2017 and 2018 dengue cases in Brazil, and I think through much of South America, were reduced by as much as 75%. And the question is why? Well, Zika, in some senses, behaves like a fifth dengue virus. Now, dengue virus is, if you acquire a dengue infection when you have no prior flavivirus immunity, you have a primary dengue infection. And that leaves you at lifetime risk to experiencing a secondary heterotypic dengue infection, enhanced infection. So dengue infections and susceptibles leave those people at risk of enhanced dengue disease. And Zika behaves in the same way. So Zika infections of susceptibles can be expected to leave them at risk. Now, the full extent of that at risk across 4 different dengue viruses, I don't think we fully understand. The other attribute of dengue is that if you have a single dengue infection and are infected with a second virus, this is a second heterotypic infection. That's the infection that places you at risk clinically, but serologically and immunologically, that infection actually protects you. So you now develop an array of antibodies that protect you from clinical disease with a third and fourth dengue infection. So presumably Zika must do somewhat the same thing. And then a final question that I've asked on this slide is, do Zika antibodies cause dengue ADE? And if so, is this massive increase in dengue cases that are reported in Brazil this year, is that an example of Zika antibody-dependent enhancement of dengue? Next slide. For those of us who worked in the old Walter Reed Institute of Research building in Washington, D.C., that's the famous global map that was put up probably during World War II. This is an institution that started during World War II and created a mechanism of research funding which is really very innovative and is now disappeared. But I think everybody working on dengue needs to think about it, because why, if I went to Thailand in 1962, this is now, what, 62 to 2024, 60 years. Why have we gotten so little distance in control of dengue in 60 years? What's going on? Well, part of it is that a lot of research sort of directed at the lab problems rather than the epidemiology problems. So I'm in favor of redirecting research in a coordinated fashion so that we could actually identify what are the really important problems, and let's attack them until we get them solved. So with that, I'll quit. Sorry to take so long, but it's been a pleasure to talk to you.
Okay, so Adam's in the control booth. Oh, there he is. Hello, Scott. How are you? It's Stephen Thomas.
Hi, Stephen.
Thank you very much for being with us. We really appreciate it. We have about 10 minutes for Q&A, so are you willing to take some questions from us?
I am. I'd like to say hello to all my friends in the audience who I haven't seen for a long time. I'm very sorry. My apologies for succumbing to something called old age, but I'm here, so I'm happy to discuss. I apologize for just adding bleak reviews of the dengue situation after 62 years, but we're all in the same game, so we'd like to make things better. So I'm here to talk about making things better.
Great, thank you. Any questions? Who wants to start? Way to go, Louis.
Scott, it's Louis Macario. Thanks for being here. Maybe give you an opportunity to elaborate on the data that you were mentioning before the talk from Bangkok Children's Hospital that maybe we're not, we haven't been taking into adequate consideration.
Were you able to hear that, Scott?
Well, I'm not sure I understand the question, but I would like to add some information that I think is important to everybody in the audience, and that is, what is the biological response, that is the clinical response of human beings to dengue viruses. And this needs to somehow be drummed into everybody who works on dengue. It isn't intuitive. It's in the literature, but boy, you have to dig it out. I can see it's really difficult to find. But let me just tell you, there are 4 different dengue viruses, and you all know, and they each can infect human beings. Human beings, differing by ethnicity, have somewhat different responses that we talked about that in the lecture. But the critical thing is the sequence. 4 different dengue viruses produce primary dengue infections in humans, but humans come small and large, and children have mild primary dengue infections. The most severe is dengue 1 and dengue 3. But adults universally have overt dengue infections, sometimes quite severe. And if you're like me, have multiple chronic diseases, and you have even the first dengue infection of your life, it could take you away because it's a fairly pronounced systemic infection. Now, then dengue comes in this very unusual variety of a second dengue infection. 1, 2, 2, 3. Now they come in 16 different combinations, and all those combinations actually occur in nature, some much more frequently than others. But it's really important to understand that the outcome of those 16 different sequences of infection are not all the same. The sequence ending in 2, 1, 2, 3, 2, 4, 2, is the most severe of all. And this is documented well in prospective studies. The next sequence that's most damaging is 2, ends in 3. So 1, 3, 2, 3, 4, 3. And then 1, and finally 4. But interval is critical. Now, we had a paper earlier this afternoon which was attempting to explain what happens in children. When I arrived in Thailand, the modal age of admission to the hospital was 4. And that meant that a peak number of children, more than 50% of 4-year-old children, had already had 1 infection and were now, in the current outbreak, experiencing their 2nd infection. Now, today, the modal age is maybe 15. And what's happened is that gradually, as Thailand and all the other countries in Southeast Asia and even throughout the world have improved their habitation and combated to some extent Aedes aegypti, reduced Aedes aegypti breeding, the virus infection rates have steadily fallen. So now, as I said, the modal age may be 12 or 13. Now, I actually calculated back in the days when the modal age was 4, that the average annual dengue infection in that period was 15% per year. And if you get to modal ages of 12 or so, you're down around 5 to 7%. So what's happened is there's been a steady decline in dengue infections. Well, why is that? That's a steady decline in mosquito populations, a change in housing. When you build 30-story air-conditioned buildings, you don't give Aedes aegypti much of a chance. So, and you can take that all the way around the world. I object to people who link dengue to global warming. I think that sent exactly the wrong message. The message you ought to send is dengue is related to the absence of piped water. Because dengue is a mosquito-borne disease and mosquitoes breathe water. And if you don't have piped water, you have to go out and get water and you have to store it. So the whole basis of dengue is a disease among 4 billion people is piped water, is the absence of water in the household. So people store water and succumb to the bites of Aedes aegypti. So the other thing is that the interval between the first infection and the second infection has a great deal to do with outcome. Short-term intervals of months usually result in nothing. Short-term primary dengue infections are quite protective, but over a period of years, in Cuba, we were able to study intervals of up to 30 years, and this was shown in my slides. The severity of disease just keeps increasing as the interval. This probably reflects the heterogeneity of the immune response. The less effective neutralizing antibodies are that are directed at the incoming virus, is a greater the damage to the host. Another feature of dengue that I think everybody should understand is that the antibody-dependent enhancement phenomenon, it produces more severe disease in women than in men. One of the very few diseases in the world. Now, a lot of these things I'm saying because absolutely nobody either recognizes it or is studying it. What is this long duration increase in the pathogenicity of dengue? What's it due to? And then to get back to NS1, I noticed that people in the audience are sort of making doubtful statements. Well, if NS1 isn't the primary cause of dengue hemorrhagic fever, dengue shock syndrome, what is? And if there's reason to be doubtful that NS1 is the primary mover in propagating this disease, we need a larger infection attempt, research effort to tell us what the mediators of infection. My friend Ernesto telling us about complement. Well, that brings me back to a long time ago when Phil Russell and the guys from the Scripps Institute came out to Thailand and did those complement studies. But we ultimately had to settle for the fact that there was something going on that was actually attacking the endothelial cells. And now we've had this, I think, remarkable breakthrough toxicity of NS1. Anyhow, I apologize for spending so much of your time talking on.
No, thank you very much, Scott. We appreciate that. That is our time. So if there are any other questions? Yes, Young, please.
Hey, Scott, this is Young. Pity we can't meet, but thanks for giving a talk anyway. I'm curious to know more about that that dengue 4 that you passaged in primary dog kidney cells that became attenuated, but then reverted after you passaged in fetal rhesus lung. So my question is, why did we do the same thing for dengue 3 then? If we had passaged it in primary green monkey kidney cells, why was there a need to passage it again in fetal rhesus lung when it had reverted with dengue 4?
Yeah, that's an important question, and I'm sorry. Just remind me again. It slipped from my mind.
Yeah, the question is, when you passage your primary dog kidney dengue 4 in fetal rhesus lung, it reverted. But why did we do that again with dengue 3?
Okay, thank you. Boy, so the interesting thing is that was sort of a gain of function action. We never tested the dengue 4 PDK vaccine that had been passaged 35 times in PDK cells. We never tested that in humans, but we did test it in monkeys. We had a fairly complex virulence testing system in Hawaii. So I think the PDK 35 was attenuated. And the fascinating thing, and this happened right under my eyes as I was watching, is Rare insisted that all the dengue vaccines have the same substrate. So I was, quotes, forced to grow my PDK passage dengue 4 in fetal rhesus lung. And when we then tested that product in 5 different human beings, 3 or 4 of them actually had clinical responses. So I know that if it wasn't a reversion, it certainly the fetal rhesus lung certainly didn't help. But I can tell you guys who are all listening, that there exists in the world a vaccine that has a PDK adapted dengue 3, because the great tragedy of the not Ramatipidi vaccine in Thailand was that they couldn't get any of the dengue 3 viruses that they had available, of which there were many, to grow in PDK cells. So they just sort of gave up and then used a primate cell line. And over many, many years, the poor old Sanofi, went back and redefined the dengue 3 and put it into Hong Kong medical students, and every single one of them came down with dengue fever. So that was the end of that. But it turns out that probably the PDK passage 1, 2, and 4 are pretty good vaccines. And along comes Takeda, and he grabs the dengue 2, which actually started in my laboratory and ended up at CDC in Fort Collins. And if you look at the data that Takeda has given us, the one component of their vaccine that really works is the dengue 2. So that's why I put in my lecture that, boy, maybe we need to work harder on trying to find PDK passage dengue viruses. And there is one in our old friend in Thailand, Dr. Suti, has it. So those of you who are bold and brave should get a hold of that guy, because there actually is a tetravalent PDK passage dengue vaccine. It's available, but it's in the hands of a very, very shy guy who's not going to make any effort to get. He is making an effort, but he doesn't have the authority and the resources to get all the way over the hump.
Well, Scott, we greatly appreciate you joining us today and sharing your experience and your perspectives with us. Thank you very much.
Okay.